Okay, why don't we get started? Thank you again to everyone for joining us today. We are going to be giving a little update on our production release, going through some of the events that we have upcoming. And then my colleague Julie Jurgens here will be presenting a new feature, which is the Cell State and Program Explorer, which is powered by Liger. So be on the lookout for that coming soon. So first off, I just want to walk through some of the events that we've had recently and also upcoming events in the next few months. So first off, thank you so much for the NIDDK for including us in the Data Science Corner as part of the American Diabetes Association conference. This was a fantastic event. It was great to meet all of, the people who were at the conference, but also to meet with colleagues that, we know and love. So it's just a great opportunity. As you can see here, we had a really great presence at the booth, and we had around over 200 people visit. So that was very exciting for us. This event was, one of our key events that we go to every year. So we greatly appreciate if you came by and stopped to see us, or if you came and stopped by our colleagues at the All of Us program. We're very excited that we had such a successful event. And as part of that as well, props to Julie. Julie gave a presentation on the human islet cell identity and organization, which was a sneak preview of what she's going to be sharing a little bit about today. So don't worry if you weren't able to attend, you haven't missed out anything there. So as part of this, we have other events that are upcoming for conferences. And so our next one is part of the Common Fund data ecosystem, as we are funded to be the Knowledge Center. And so as the Knowledge Center, we will be attending the Intelligent Systems for Molecular Biology Conference in Washington, D.C. this year. And this is going to be July 12th through 16th. Again, Julie is going to be presenting on behalf of our organization. She's going to be giving a short talk, the Common Fund Data Ecosystem Incubator for Pan NIH Resources, Enhancing Interoperability. And so this talk is going to be during the Bosch track on July 15th from 2:40 to 3 p.m. And then she's also going to be presenting this same information at the poster session. So please keep an eye out if you're going to be there. I also want to highlight there's going to be several other programs from the Common Fund data ecosystem. As you can see on the right here, there's Biomarker KB, the CFD Connect, so our Integration and Coordination Center, the Workbench, Data Distillery Knowledge Graph, Safety Training Center, GlideGen, and Kids First. So if you have any questions, concerns, or you're interested in learning a little bit more about the programs of the Common Fund data ecosystem, you can learn a little bit more at this conference, which we'll be attending. And if you're unable to attend that conference, we'll also have our Common Fund Data Ecosystem public webinar series continuing this year. And so our next session is going to be Friday, June 26th from 1 to 2 p.m. Eastern. And Cell Maps for AI, or CM4AI, will be presenting, and they'll be sharing a little bit about the data generation project, which seeks to map the spatial temporal architects of human cells and use these maps towards the grand challenge of interpretable genotype phenotype learning. So if you are interested in learning a little bit more about them, please feel free to join us on the 26th. We're very excited for them to be participating in this event. We also have several other webinars the last Friday of every single month from here until November. So again, you can learn a little bit more about the Common Fund data ecosystem. I also want to highlight a funding opportunity that is available through the Common Fund, just in case this is of interest to anyone who is on the webinar today. And so if you scan the QR code to the right, it'll give you some information about this funding opportunity, which is pilot projects enhancing utility and usage of Common Fund data sets. So specifically, this funding opportunity is designed because there are many valuable and widely available data sets that have been generated by multiple Common Fund programs. So the purpose of this NOFO is to announce the availability of funding to demonstrate and enhance the utility of selected Common Fund data sets including generating hypotheses and catalyzing discoveries. And so, as part of this, award recipients are asked to provide feedback on the utility of the Common Fund data resources. This application is closing June 23rd, and there's an expected 13 awards for 200,000 award maximums for this. So if you're interested, like I said, please feel free to scan the QR code as this will give you all the information about it, and it is closing in 2 weeks. Lastly, our next webinar that we'll be having for the Pink Base as part of this series is going to be July 9th. So this is in a month, and we'll be showcasing the beta release of the Gene program features. So we're very excited about that. I also want to highlight some other dates where we'll be representing Pink Base, and this will be August 27th and October 20th. So please mark your calendars. It's going to be the exact same time as these sessions, so you don't need to register if you're already attending here. You already have that registration. This is just highlighting what will be at those sessions particularly. So these are all the different events that we will be attending and will be a part of in the upcoming months. And we're very excited to be involved in all of these. So next, we're going to be showcasing a little bit about our latest production release to our knowledge portals. And then after this, we'll have Julie go into our new feature, which is the highlight of this production release. So first thing that I want to share is we've added a new option on top of the menu bar for all of our knowledge portals, and this is called the News Hub. And so essentially, this News Hub is going to give you any information of news and newsletters that we will have throughout our knowledge portals. So if you were to click on the News Hub at the top here, you can see there's a drop-down menu that includes the news, newsletter, and blog. So one thing that I just want to highlight is our news has not changed. Anything that you would typically see for news on the homepage, you can access that accordingly. And so you can also select it from the drop-down here. But first, I want to showcase our newsletter. So if you select newsletter in the drop-down on the homepage here, it will take you to our KPN newsletter, which is a brand new section of our knowledge portals. And so these are going to be released quarterly. These are going to showcase updates from production releases, collaboration opportunities, funding opportunities, and several upcoming events that we have. So anything that you need to know about the Knowledge Portal Network will be located here. As part of this, we'll also have this archive, as you can see here, which you'll be able to access all of the other newsletters after they are released. And so the idea is you can come back and search this anytime you'd like. And so we're really excited to have this feature added as part of our production release. And so the other item that was under the News Hub tab here is the blog. And so this is a brand new feature as well that we're extremely excited about. And so this is another page here where we're going to be showcasing some of the blogs written by our colleagues. And the idea that we had specifically about this blog is that it would be able to share scientific research, new insights and perspectives that our team and beyond will showcase. And so the idea is we'll have, as you can see the example here, this is from our colleague Chung. These will be updated frequently based off of, different things as they come up. So for example, our goal is to make complex ideas accessible, meaningful, and relevant. So this is, going to help for people who are maybe, a seasoned researcher early in their career, or you're just curious about the world of genetics and genomics. So we're aiming to highlight new discoveries, evolving trends, and just essentially provide a space for thoughtful commentary. And so, as I mentioned, this is brand new, but if you're interested in learning a little bit more about our first blog features Trong, one of our colleagues who you may know, and it is titled The Bottom Line Association's A Scientific Saga. And so in navigating this page, if you're interested in reading more about this blog, you can click on the read more and it will take you to the blog itself. So I encourage you to go take a look at that. Like I said, we have a couple new blogs that are going to be coming up shortly, and we're very excited to launch those to the public. So keep an eye out. And if you aren't sure if there's going to be new blogs out, we will be promoting them on our socials and also as part of news items. So we'll be able to keep you informed as much as possible with these new releases. So next, I'm going to be talking a little bit about our data ingest across our portals. So in this latest production release for the Common Metabolic Diseases Knowledge Portal, we've added in over 1,000, or we have 1,000 phenotypes. And so here we've added 90 new ones to make that total again 1,040. As part of this as well, we have added 146 new genetic data sets for a total of 919. And we've also added in 676 new bottom line attack-seq data sets for a total of 7,299 genomic data sets. And you can explore these in the CMDKP. If you go to the datasets page, there'll be information on the ingested data. And we're really excited to continue to expand our resource in this way. For the A2FKP, we've added 96 new phenotypes. So now we have a total of 1,482 that are represented. We also have 161 new genetic data sets for 1,267 total. And then again, we have also added in the same amount of the attack-seq data sets for a total of 7,299. And so I want to dive into this data a little bit further. So of this ingested data, we've actually received a significant amount of new reproductive and sleep and circadian traits. And so as part of this, we're constantly looking for different areas in the data sets. So in this specific area, we're really excited to see some of this growth. And while this is not the comprehensive list of new traits, these were a couple across all of the data sets that I thought were quite interesting. So just some of these include hearing loss, sitting height, aortic valve area, asthma, scoliosis, infertility in both male and female, caffeine consumption, device at bedtime, light exposure for summer and winter, nap duration, nightmares, sleepwalking, and time zone travel, which I think a lot of these are relevant to, things that we may face in our everyday lives. So I just find that really fascinating that we have some of this data now. And again, you can explore all of this new data on the dataset page on the A2FKP, and we encourage you to do so. So with that, that's a little bit more about some of our new updates to the menu and the data. And so now we're going to be walking through some of our brand new features, and I'll be handing it over to my colleague, Julie Jurgens, who will be showcasing a little bit more about the cell states. So thank you, everyone. And Julie, I'll hand it off to you.
Thank you so much, Mackenzie. All right, I'll go ahead and share my screen here. All right, can you see it okay?
Yes, that looks great. Thank you.
Great, thank you. So, hi everyone. For those who don't know me, I'm Julie Jurgens. I'm a colleague of Mackenzie's and a representative of the Knowledge Portal Network with Noel Burt and the Knowledge Portal team at the Broad Institute. So, I'm going to tell you about a new feature that we've just added, released last Friday, for identifying diabetes and other condition-relevant gene programs across metabolic tissues in the Common Metabolic Diseases Knowledge Portal. So have you ever asked the question, what cell states and gene programs are my favorite gene involved in, and how do these relate to disease? If you have, the feature that I'm about to show you will help you to address those questions. We've made this available on our website, which is cmdkp.org. And for those who are unfamiliar, our Common Metabolic Diseases Knowledge Portal, or CMDKP, is an open, NIDDK-funded, community-driven web portal that connects genetic signals to common metabolic diseases. So we got our start in type 2 diabetes, and we've gradually expanded to accommodate all of these other types of diseases and systems. Our bread and butter is sort of genetic and genomic work. So we aggregate various genetic association data, do meta-analyses across those using the bottom line analysis, which Mackenzie just told you about. So if you're interested, you should read that blog and check out Tron's paper. And we also bring in functional genomic annotations through our colleagues at the Common Metabolic Diseases Genome Atlas, or CMDGA, with Kyle Galton's team at UCSD. We couple these with various bioinformatic tools and methods that we run across the data, as well as curated community knowledge to allow our users to ask these questions. So what genes are involved in the risk of the phenotype that I study? And does the gene that I study have a role in human disease? So as Mackenzie mentioned, we now have over 1,000 phenotypes represented in the CMDKP for over 900 genetic data sets, over 7,000 genomic annotations in various disease-relevant tissues, have run 10 bioinformatic methods, and importantly, have engaged with the community to understand what is important to them and how we can leverage their expertise to boost the knowledge that's relevant to a given topic. So one feature that we made available a few months ago is our CMDKP single-cell browser. This features integrated single-cell maps for 9 metabolically relevant human tissues to date. This is available on the KP Labs drop-down single-cell browser if you navigate to our cmdkp.org. These maps were developed by Kyle Galton and his team, who you can see down here, and then implemented by our team, who you can see here. To date, we have these 9 tissues represented: artery, heart, hypothalamus, kidney, liver, skeletal muscle, pancreas, and white adipose, subcutaneous, and visceral. You can see that each of these aggregates data from multiple sources and a large number of donors, in some cases with over 1 million cells represented. And some of these are the largest integrated maps available for these tissues to date. So this is just a sneak preview of what this feature looks like. So again, if you navigate to KP Labs, single cell browser, you can see the interface. So the interface starts with interactive UMAP plots, where you can see cell type clustering for various cell types that are available in the selected tissue of interest. You can then query a gene and see where it's expressed within those cell types. You can also color by various other fields that you find important, for instance, by data set or age, and see gene expression by those variables as well. Scrolling down on this page will give you an interface that looks like this. So here you're seeing the proportions of various cell types within the data set. So you can see in this particular data set, smooth muscle cells are the highest represented. And for the gene that you actually queried above in the UMAP plots, you can see violin plots of gene expression. You can also get these plots for various other fields that are of interest. For instance, let's say you want to stratify by body mass index or by age. You can select those variables as well and see expression of your gene by those traits. Finally, the bottom of this interface shows the top marker genes. Here we're looking at all cell types represented in the data set, but you can filter just for a specific cell type if you wish to see that as well. All these charts are downloadable. So now I'm going to tell you about the brand new feature that we just introduced last week, and that is gene program inference. So this work was led by various individuals on our team and on Kyle Galton's team. So Jason Flanick, Kyle Galton, Patrick Smadback, and Alex Schillen, DK Jing, and Wei Huang. This interface leverages a technique that we call non-negative matrix factorization, or NMF. This is a dimensionality reduction technique that identifies latent structures, or what we call factors, in high-dimensional data. So here, if you're looking at an original matrix, You can simplify that into submatrices and pull out latent structures or, hidden sort of information and patterns within the data. So this type of a technique has been used in a number of different settings, some relevant to biology and others not. And we'll give one example that is from outside of biology here. Sorry, just a second. There we go. So here, this analogy we like to use is for movie recommendations. So let's say you're like me, you're hopelessly addicted to Netflix, and you're constantly trying to wean yourself off or cancel your subscription. Just as you're about to do that, Netflix always seems to email you with, something like, "Have you seen Love is Blind 3? Did you know that's on Netflix?" And then they rope you right back in, right? How do they do that? How do they know exactly what you like to watch and, keep you subscribing? This is one way to do it. So here you have a matrix of users by movies. You can simplify that into two submatrices, one for the users and one for the items or movies. And in this case, NMF analysis will derive the factors. So in this case, those are the preferences for movie viewing from those submatrices. So by doing that, they can see what other users who are like me tend to watch and get an idea of what I might like to watch as well through inference based on the data. On the CMDKP, we've done something very similar. So in this case, NMF analysis derives the factors. In this case, those are coordinated gene expression patterns, or what we call gene programs. So those are just groups of genes that reflect some sort of underlying biological process, cell state, or regulatory mechanism. Here we're starting with a matrix of genes by cells, simplify that down into a gene matrix and a cell matrix, and use that to infer the gene programs or groups of genes that are relevant to some sort of biology within the data set. As we were doing this analysis, we realized that it would be nicely complemented by a second type of analysis, and I'll explain what that is. So the analysis I just described is for gene programs, right? These are informatically inferred analyses for groups of genes with some sort of a coordinated expression pattern in a cell type. Because they're informatically inferred, this is a data-driven approach, and it's very exploratory, right? So the idea is that you can pick out patterns that you may not otherwise see, or that may not have been previously reported, because they're strictly based on the data. However, we thought this would be nicely complemented by something based more on well-established known biology, and those are what we call cell states. So those are transcriptionally defined cell populations that are curated from sources like published literature, published sources of marker genes, and they capture some sort of known biology. Collectively, these gene programs and cell states can be used to understand how a cell type, can um, inform human disease and how changes in those cell types can be linked to human disease. So how is this different from what existing resources typically offer? So existing resources usually offer single cell atlas exploration of various types, and they do this quite well. They also offer various post-GWAS cell type and gene set enrichment analyses. Now, this feature is unique in that it connects harmonized healthy and disease single cell maps across key common metabolic tissues with a number of things. One is the curated cell states. Second is the informatically inferred gene programs across multiple tissues at scale. Systematic comparison between those cell states and gene programs so you can see how they complement each other and use the cell states for validation in some cases. We also provide links to genetic support for metabolic traits and diseases, and I'll explain how we've done that. Finally, and most importantly, these analyses are all publicly available and browsable through pre-computed analyses and visualizations. So again, we have this for all 9 tissues that are available within our single cell maps and have almost 600 curated states and 1,400 inferred factors. You can access this on the CMDKP through the KP Labs feature and the dropdown that cell state and programs. That'll take you to an interface that looks something like this. So to start, you'll be prompted to search your gene of interest. So here I've searched for PNPLA3, and then it'll ask you to select a tissue and cell type of interest. So here I've selected liver and hepatocyte. And that'll take me to the next part of the interface. So here we're looking at a side-by-side panel that shows the cell states next to the gene programs. The cell state component provides those curated marker-defined biology and gives you information about expression of each of those cell states within the selected cell type. To get more information on how these were calculated, you can hover over either of these headers up above and see what these actually mean and how they were derived. You can also hover over any given row to get information about that specific cell type, cell state, with a description and the marker genes that are pertinent to it. And clicking on that actual row will take you to a much more detailed um, metadata description with more information about the marker genes, specific references from which they were curated, and then other information. So the programs, the gene programs that are most relevant to it, and then human genetic traits that are linked to it. On the other side of the interface, you can see the gene programs. So again, these are the computationally inferred latent factors that we got from NMF, the non-negative matrix factorization technique that I just described. And we've derived these using a method called Liger. So here you can see each gene program, again, its expression. You can hover to get similar information to the cell state component that I just showed you. And again, clicking on any of these rows will take you to detailed information about that gene program, um, the curated states that best match it, the top genes and their relative loadings within that, and then the traits to which they anchor. Further down on the interface, we have this correlation matrix showing the gene programs here on the x-axis and the cell states here on the y-axis. And this shows how well these are correlated. So here, lower correlation is in red, higher correlation is in blue. Um, this is a new analysis, and again, to our knowledge, this has not been done systematically at scale to date. So we're in the process of validating these now. But we've gone through and spot checked a couple of examples and see some reassuring instances where the cell state and gene program correlate as we would expect. So in this example, hepatocyte identity is both a cell state and a gene program, and we can see those are positively correlated. Next, you can select the dropdown up here at the top to observe various other types of relationships between those states and programs. So if you want to look at marker enrichment across those, you can select those and see coloring by that as well. Now, finally, the bottom part of the interface links this cell state and program analysis back to our bread and butter, which is the study of the genetics of human phenotypes and traits, in this case, relevant to common metabolic disease. So here we're seeing results from priors inferred from gene annotations, or PIGEN. This is a statistical genetic method developed by Jason Flanick and his team. You've likely heard some of our other webinars about this, but if you have not, you feel free to check out our website and you can get the link and learn more about that as well. So here you're seeing the results from those pigeon runs for both cell states and gene programs here on the x-axis, and on the y-axis you're seeing various traits. So here you can see in this example for the query gene PNPLA3 in liver hepatocytes, there is significant genetic linkage to these two cell states or gene programs for bile acid transport metabolism and for hepatocyte identity, and for various lipid traits. This is consistent with what has been reported in the literature for PNPLA3 and its connection to these various lipid traits, and provides some insight into how that might be happening. So in summary, gene program and cell state analyses are two complementary approaches for understanding cellular function and its roles in human disease. The CMDKP connects these harmonized healthy and disease single cell maps across key common metabolic tissues with curated cell states, informatically inferred gene programs across tissues and at scale, compares the cell states and gene programs, and provides genetic support for the metabolic traits and diseases. And all of these analyses are publicly available on the CMDKP through this first of its kind resource. So as Mackenzie mentioned, we were fortunate to be a part of the NIDDK Data Science Corner at the American Diabetes Association meeting in New Orleans this past week. And there we, along with various of our colleagues, showcased a number of open resources funded by NIDDK for science. So I told you about the CMDKP, but we have various other resources like our Mammalian Adipose Tissue Knowledge Portal, or MATKP, PancBase for type 1 diabetes in the human pancreas. Um, and those are resources that we've developed, and various of our colleagues have also developed some wonderful open resources that you can check out here too. So with that, I would like to thank the incredible team who put so much work into making this happen. That I'm just the person presenting this, but a lot of work went behind the scenes to make this happen. And we are hugely grateful and excited to be showcasing this resource. So thank you to everyone in the BERT Lab, the Flanic Lab, Kyle Galton and Ben Voigt's teams, to the NIDDK and FNIH for their support, and to all of you for your attention. So with that, thank you, and we're happy to take any questions.
Thank you, Julie. So I do see one question that is in the chat. So when are we at other conferences? Um, so we will be at ISMB as part of the Common Fund Data Ecosystem, and that will be July 12th through 16th. We'll also be presenting the Common Fund, sorry, the Common Metabolic Diseases, too many acronyms of the similar kind. And that will be as part of ASHG in Montreal in the fall. And we'll also be showcasing the A2FKP and the CFDE as well. So we're going to be at all of these different events. We are very excited to be, at all of them. Again, we hope to, meet with all of you and showcase all of our information for new production releases. There are ancillary sessions that we are going to be applying for ASHG as well. So if you've been to ASHG in the past, we have had those sessions, so we will give you those details as soon as possible. But we're really excited again to be, doing as much as we can with the community and then also speaking with all of you. So we're really looking forward to it, and then keep an eye out for us at those events. And then ISMB, if I didn't mention this, is going to be in Washington, D.C. So we'll be traveling a lot for all of these events, so hopefully we'll be able to see new faces as well. Great. Any other questions regarding any of the single cell browser, the cell state gene program interface, or any production release items? Well, if not, Julie, I know we've talked a little bit about, what we've produced, and you've kind of showcased a little bit of where we're to go. But what do you think is the next phase of this for our knowledge portals?
Yeah, that's a great question, Mackenzie. So I think what I just presented is kind of the beginning. There are a number of different directions that we plan on taking this. So the first is by continuing to improve these single cell maps by integrating data from additional sources. That's something that Kyle Galton and his team are actively working on to, build out these capabilities even further. Um, a second thing is, validation of some of these results, right? So as I mentioned, that's to our knowledge, this has not really been done systematically. So right now, we've put this up. We want people to play with it, but many of you are often the experts in your given cell type or disease, and we would love to, have you play around with it and let you know what have let us know what you think, basically. So um, I think that would be hugely helpful, so we can think about how we can build this out and make it more useful. We actually got a fantastic question at ADA about this, which was kind of related to that. It's from somebody who was an expert in the human pancreas, and they took a look at that map and they said, well, this is a great start, but there's a lot that's missing from the curated cell states, right? So one thing I didn't go into during the presentation is that right now these curated cell states are based on an AI assisted workflow, right? To bring in well curated marker genes from various um, of publications identified by those systems. However, in some cases, you all probably know the literature much better than we do, right? And if you take a look at some of those things, you can say, okay, this is something that I think is well vetted. This is something that, subsequent literature has expanded our knowledge of or changed our knowledge of. And so in some cases, we think this would actually benefit from engagement with all of you to do that. So we're looking at ways to do that more in a more structured way, and maybe build out some sort of a forum for folks to comment. But in the meantime, feel free to email us through the link that Mackenzie just provided, and we'd love to talk to you.
Awesome. That's fantastic. Thank you, Julie. So we have a couple questions in the chat, and the first is from Eve. And so Eve said, not sure if you have anything on the HPA axis. That's part of the major hormone system driving weight. It seems to respond to light exposure. And then if there's no answer on that today, that's okay. But that was just a comment slash question from Eve.
Hmm. Eve, that's a great question. Thank you. I can't say offhand if we do, but this would be a really interesting case where we could, take a look, and have you take a look at it based on your expertise and your investigations into this. So you say, okay, relevant to the circadian system as well. Great. So right now, this interface allows you to query by a gene to start with. So instead of querying by, the circadian traits or whatnot, I would encourage you to start by finding genes that are involved in the HPA axis that and to use the interface to search for those. Um, and then once you've done that, you can select the tissue type and cell type that you think are most relevant to that axis. So, whether that's hypothalamus, that's one that we have in there, or something else, I'm not sure, but give that a try, and we'd be curious to see if that pulls out some of the expected associations that you'd be expecting. That's a great example. Um,
Great.
and yeah, as I mentioned, this is largely exploratory. So if this is a case where, you haven't found a lot of research on this to date, this might be a case where you can kind of take a look and see what it pulls out to generate new hypotheses. So
Thank you,
thank you.
Julie. So there's another question in here, which is where to find which method is used for the single cell RNA analyses and cell annotation method used in this pipeline.
So that is a great question. These analyses were done by Kyle Galton and his team. That I think you pointed out, we actually don't have that sort of explicitly stated on the website as is. So that's a great point. We'll work on getting that up on the website so that you can take a look at it and get more details on that. Thank you.
Great. Thank you, Julie. I'll give it another minute or so to see if we have any other questions. Like I said before, please feel free to use the chat, the Q&A, or if you want to ask your question live, you can raise your hand and I can take you off of mute. But again, it's if you have questions that come up after you've been exploring it a little bit more, we'd be happy to, answer any questions over email as well, because I'm sure things will come up as you're using it. And so again, as Julie said, we really do want that feedback and any perspectives that you might have. So I'll give it another second to see if we have any other questions on that front. I think one question that I might ask to the group while we're waiting for any other questions to come in is if there are certain tissues that you would like us to look at next. We also want to hear from the community in terms of what's relevant to your research. So if you have any thoughts about that, we'd be happy to, note those down as well. So keep that in mind as well.
One other thing I'll say while we're waiting, Mackenzie. So in terms of future directions here, I've shown these analyses applied for this particular resource, but we have other resources that I mentioned that we are developing out. So we're looking to do similar analyses for data sets within those resources. So two resources that we're looking to bring this to are the MAT-KP for mammalian adipose tissue, to PancBase for um, the pancreatic islets. And then we would actually, um, in the future, be building out other resources, which we have not yet, made publicly available, but just as a teaser, within the Common Fund data ecosystem, we're actually working with our colleagues, Shankar Subramanian at UCSD and Avi Mayan at Mount Sinai to build out a liver resource. So that is not yet publicly available, but we're actively working to build this out. Um, so once we have that ready to go, we will release that and would love to get your take on that too.
Great. I think we have a lot to look forward to, which is very exciting. So, since I don't see any other questions, I think that's a great note to end on. We will be showcasing all of our updates, like we said, throughout the rest of the year as we make them. As Julie was saying, we have a lot to look forward to, specifically for this resource and others. And please keep an eye out for us at the different conferences. And we look forward to speaking with you a little bit more at each of those. So actually, one question I do see is from Eve again, the liver is very complex. Can we divide it out via cell types and function?
Yes. So on our existing interface, you can do that by selecting a cell type to start, not by function, but by cell type within the liver. And for a given gene, you can get information about that. Within the liver resource that we're building for CFDE that I just mentioned, that's actually something that um, our team and our colleagues within the Data Resource Center from CFDE are actively working on building out in other ways. So, exploring different cell types, what their roles are, and such. So stay tuned. That'll be coming soon.
Great. Thank you for some clarity on that, Julie. Um, okay. Thank you again so much for presenting our brand new resource. As we keep saying, please feel free to play around with this feature and others, and also explore those brand new data sets. We're very excited to have, all of that included in our production release. And again, we'll be continuing to update as we go along. So thank you so much, both to you, Julie, and for everyone who attended today's session. And we look forward to seeing you all soon. So thank you. Have a
Thanks,
great rest of your
everyone.
afternoon.
Bye
Bye.
bye.